I'm grateful to be a part of this session and to open a conversation that brings together scientists at many different stages of their careers that feels especially meaningful in the context of the larger forum, which reminds us that the challenges facing humanity today, from global health and climate change to environmental stress and geopolitical division, do not respect borders, disciplines, or generations. One reason science has mattered so much in moments like these is that basic science gives us a shared way of understanding the world. It offers a common language grounded in evidence, transparency, and debate that makes cooperation possible even when agreement is otherwise hard to find. The researchers we'll hear from today represent 10 promising directions, but they also represent something more personal to many of us in this room. That moment in a scientific career when curiosity is still wide open and the future feels full of possibility without limits. Choosing to work on problems whose answers are uncertain and whose rewards may lie far ahead takes patience and courage. It is the work that sustains science over the long term. To our laureates and senior colleagues, thank you for being here and for engaging directly with the work. Your questions, perspectives, and examples shape not only individual projects, but the standards and values that younger scientists carry forward. And to the younger scientists presenting today, this is not about having all the answers. It is about sharing how you approach problems, what motivates your questions, and where you see both promise and uncertainty. Those are the conversations that move science forward. With that spirit, let us begin part one with openness, generosity, and a shared sense of responsibility for the future we are all helping to build.
Today, I'd like to talk to you about how two physical phenomena, the quantum Hall effect and superconductivity, can be merged together. And we found that graphene offered a platform to merge these two effects. Now, if you take a superconductor, a current can be carried without dissipation by Cooper pairs inside the superconductor. These are quasiparticles that carry twice the charge of an electron. And if you bring a normal metal in proximity to this superconductor, the Cooper pairs may leak inside this normal metal and induce a supercurrent at a distance limited by the superconducting current's length. The Cooper pair may leak inside the normal metal at distances limited by the superconducting current's length. Now, if you make a normal metal to superconductor junction, you can induce a flow of Cooper pair from one superconductor to the other if you take two electrons in the normal metal and they merge at the superconducting to normal interface at distances limited by the superconducting current's length. The quantum Hall effect, on the other side, is a different effect that emerges when you take a two-dimensional electron gas and you apply a strong magnetic field. In these circumstances, the field forces electrons into quantized cyclotron motions, which are the Landau levels. Now, if the Fermi energy lies between these levels, the bulk of the material becomes insulating, and the current can be carried by edge channels that emerge. The electrons in these edge channels move only in one direction and without scattering. Now, combining superconductivity and the quantum Hall effect has sparked a lot of interest since the 1990s, with a motivation that is threefold. First, this hybrid system could force the electrons in the edges to pair up, which would host non-abelian ions, for example, like Majoranas in the integer quantum Hall effect regime or parafermions in the fractional quantum Hall effect regime. And these could be useful to protect information in quantum computers, for example. Secondly, such devices could close the metrology triangle. We currently have a standard for the volt through the Josephson effect and a standard for the ohm through the quantum Hall effect, but we lack a fundamental description for the ampere. And a perfect interface between a superconductor and the quantum Hall edges could provide a way to achieve the third leg of that triangle. And thirdly, quantum Hall edges are ballistic and therefore can provide a platform to study phase coherence over long distances. And one could imagine, for example, creating artificial molecules that way. The problem has been looked for ages, and there were many efforts to combine superconductivity and the quantum Hall effect regime. And to achieve this, one would, to achieve clean and well-separated edges, you would need high magnetic fields, and in these conditions, the magnetic field, the superconducting current length would scale as 1 over the critical field. To couple these two counterpropagating trajectories with the superconductor, you would therefore need a superconductor with a low critical field, which would destroy the superconductivity in the system. So there's a fundamental conflict between achieving a supercurrent and coupling two quantum Hall edges. There were many efforts to combine the two, and so far the best devices were very narrow edges, where state-of-the-art devices are microns wide and achieve supercurrent in the order of 0.1 nanoamp. This is very fragile, very small, and brings additional difficulty to achieve topological superconductivity. We tried different strategies, and our strategy was to couple two edges at the center of the device. We hoped that they could couple more strongly to superconductors that way. We explored several approaches. The first one was etching a line, a very thin, narrow line in the center of graphene devices to couple counter-propagating edge states on each side. This was unfruitful. We tried another strategy to use wrinkles naturally occurring in graphene, which was, again, unsuccessful. And we tried to achieve steps in the substrate, which, again, was unfruitful. Eventually, we realized that we needed a system where two propagating quantum Hall edge states would coexist on the same atomic lattice, which would preserve the phase coherence in the system. And we found another system which was offered by bilayer graphene. If you get a graphene bilayer, you would realize that in some regions atoms sit on top of one another, while in other regions they do not. And this symmetry, this is not entirely symmetric, and the symmetry can be reversed. Now, if you peel off two layers of graphene from one another and you stack them together again, what you'll end up with, because of strains and defects naturally occurring in the graphene, is large domains of locally aligned regions separated by very narrow domain wall. And what's interesting here is that the stacking is continuously and smoothly evolving between one stacking arrangement and its opposite through a narrow domain wall. This provides an interface that is perhaps 20 to 50 atoms wide, and the topology is reversed between each domain, which means that there are actually one-dimensional channels that propagate through these domain walls. And this domain wall provides exactly what we need, a one-dimensional conductor that can hybridize with superconducting quantum Hall edges, or with quantum Hall edges, sorry. So we tried to do this, and we achieved devices. We located the domain walls, and with the lithography created superconducting electrodes. We can now turn on the magnetic field to enter the quantum Hall effect regime. What we observe here at 3 Teslas is we measure the resistance as a function of the applied current. We see a region of 0 resistance immediately followed by a peak in the resistance at a critical current. This critical current is about 20 nanoampere, which is about 100 times larger than what was previously achieved using edges on opposite sides of devices. Even more striking is that when we vary the magnetic field, we can monitor the critical current as a function of the magnetic field, and we see that it doesn't vary over a field scale of a few Teslas. And this means that the two counter-propagating states in the domain wall are separated by distances less than nanometers. We can also use 2 parallel domain walls and observe the clear squid-like interference pattern. These almost perfect oscillations show us that the two domain walls carry approximately the same amount of supercurrent. And they can couple in a one-dimensional system. The superconductivity can couple with this one-dimensional system. Finally, by increasing the temperature above the critical temperature and the magnetic field above the critical field of the superconductor, we can measure the conductance of this one-dimensional system. What we find is that the conductance reach E square over h, which is the conductance quantum, at temperatures above 4 Kelvin. That means that the channel that we're working with is clean, it's ballistic, and has 4 electronic modes throughout its length. Now, in such regime, the appropriate theoretical framework is the Luttinger liquid. Luttinger liquid is a universal description of a clean one-dimensional conductor where interactions dominate, quasi-particles disappear, and electrons fractionalize into collective waves, which makes one-dimensional systems fundamentally different from 2- or 3-dimensional metals. You might think that 1D systems already exist, like carbon nanotubes, semiconducting nanowires, or charge transfer salts, for example, but that's only partly true. To observe a Luttinger liquid behavior, what you need is that the length of your one-dimensional system is larger than this coherent length, defined as the ratio of the Fermi velocity over the temperature. And if you plot this normalized by the Fermi velocity, which is a material-dependent property, you need to be above that line to be considered in the Luttinger liquid regime. And what you find is that semiconducting nanowires and carbon nanotubes, for example, they typically have very high Fermi velocities, and therefore, because of defects naturally occurring, their experimentally accessible length is very short, and it's been unclear so far whether these systems could be described by a Luttinger liquid picture. Now, in contrast, you can have quantum Hall edges, which fit very well in the system with a reduced Fermi velocity. The problem is they're hardly combined with superconductivity. And we can calculate, we asked theoreticians to calculate the Fermi velocity in the domain world system that we had, and we found that it's 2 orders of magnitude lower compared to other systems. And that leaves our system in a unique window where the Luttinger liquid physics, so the one-dimensional physics of strongly interacting system, could be explored. With that being said, before I conclude, I'd like to thank the people who've contributed to this work, all listed here, specifically Na Jin, now a professor at Zhejiang University, who did a lot of efforts in order to connect this one-dimensional system to the superconductor. And I'm also deeply grateful to Andre Geim, whose deep expertise and intuition helped shape the way we approach the problem. And to conclude, we set out to engineer superconductivity along quantum Hall edges, motivated by the search for non-nebulous anion, for metrology, and phase-coherent quantum devices, which was a challenge for a few decades. We found that graphene domain walls provide a natural platform where superconductivity and quantum Hall effect coexist in a robust way. But the most expected outcome is that this very same system gives access to something condensed matter physicists were looking for decades, which is a clean, ballistic, and long one-dimensional channels. Thank you very much for your attention.
So let me just begin by saying that the Earth is warming up. I know you all know that. We're in the process of exceeding 1.5 degrees centigrade. But the weather we see now is only part of it. The deep oceans are very cold. About 90% of the energy goes into the oceans. Half of that energy is in the deep oceans, and there's a very long mixing time. And so it'll take roughly 100 years to see the weather that we've already baked into the system because of the additional greenhouse gas effect. In order to get to, let's say, 2 degrees centigrade, you have to go to 0 emissions on all sectors of everything we do. 1.5 is already out of the picture. And we're right now emitting 53 gigatons per year of carbon dioxide equivalent. So we seem to be on track of going to about 2.83 degrees C. We're a little bit above so-called current policies. All right, so let me go back. One other thing, World Bank has suggested 200 million climate refugees by 2050. We see that 10 million refugees have transformed the politics of Western Europe and the United States. So that's another concern, because when you have failed agricultures, then sometimes local societies collapse. Okay, great stuff in renewable energies. You can get solar energy now being sold on contractual basis at 1.5 cents a kilowatt hour, cheaper than anything possible. It's unbelievable, but the news is, even though that's solar electricity, you need all sorts of other things in order to get the electricity to be useful and still have access to power when you want it. And so that means the full cost of renewable energy has to include the backup energy systems, the distribution, transmission systems, and of course, energy storage. So let's talk about electricity transmission and distribution. Europe and China especially are beginning to realize in order to take full advantage of renewables, you need very good long-distance transmission lines. The best are high-voltage DC lines, and those in the red are Europe transmission lines. And in China, you see transmission lines bringing wind resources from the northeast and northwest, hydro from the south. And so Europe and China are doing great. The United States is not doing much at all. And so, in fact, we're going to be doing, and the next 3 years we'll be doing even less. A little bit of physics on transmission lines. That's a high voltage DC line. The amount of power you can put through a line scales as V squared over R. So if you double the voltage, you can get to, for the same resistance, you can get 4 times more power. And DC lines are good. They don't have radiative losses. They don't couple to the ground. And so the efficiency, the losses are about 40% less. That's the good thing, very good thing. But even more important is you can't put AC lines underground for long distances. Anything over 10 kilometers, 20 kilometers, it does not work because of the capacitive coupling. And so you have to go DC. And if you see here in Southeast Asia, there are dreams to link Southeast Asia and to get it to Australia. If you can get it to Australia, you can have access to incredible solar power that you can pump up. So how do you get these DC lines? Well, right now, the voltages are limited by the strength of dielectric materials. They're operating at 525 kilovolts. And you say, well, why not go to thicker insulation? But as it gets thicker and thicker, it gets more and more unwieldy, and you can't do this. So there is a premium on getting better dielectric materials. So you can get up to 650 or even 800 kilowatt hours. It doesn't sound like a very sexy goal, but it's a very important goal because it will help link many places. In terms of energy storage, that's my favorite battery. The way it works is when you have intermittent renewables, you pump water. That's the energy intensive thing. When you need irrigation, you open up the valve. And so 4 or 5 years ago, 95% of all the electricity is pumped storage, but that is rapidly changing now. We're seeing battery chemical storage becoming comparable, and within 5 years, it will pass it. And so this is an example in California. We're 60% renewable energy in California. We intend to go to 85%. When you go to 85%, most of it is solar, you have a huge surplus of electricity during the middle of the day. The blue over here is hydropower. And even in the middle of the day, when you're overproducing energy, you either have to sell it or warm up the ground. You still have to keep the hydro going so the river doesn't die. And so the solution is batteries. And what California has done is that it's installed a large number of batteries to take that excess energy and use it later in the day. And as I said, China that was 16 gigawatts, which is an enormous amount. But in China, they're actually looking at order of magnitude or more increase in energy. The biggest goal is to get a battery that doesn't require conditioned environments. A sodium-ion battery can work at 60 degrees C and minus 30 degrees C. And so this is a big deal, especially for sub-Saharan Africa or South Asia, because otherwise you would need a refrigerated environment to keep these batteries working. And so if you can get these batteries to the cost of lithium iron phosphate, it would be a very big deal. They cost more, but maybe in 10 years. Batteries for EVs are remarkable. I just want to draw one thing, that this cost curve, since the year 2000, the cost has been plunging very rapidly. And if you look at 2020 to 2030, the price has been cut in half. You look at 2000 to 2020, the price has been cut by 20. And so the other thing that's very exciting is the charging rates are increasing. Once we can go out of a 400-mile range, you can go 200 miles in 5 minutes. We go back to the service station model. There's no longer any range anxiety. And that is a primary goal of these type of batteries. Well, what's going to stop it? Well, there are resources that are going to stop it. Well, we run out of cobalt, nickel, lithium. Cobalt, nickel, already too valuable. They're transitioning to iron phosphate. What about lithium? People, including myself, have figured out how to extract lithium from salty water, so lithium will not be a problem for a couple hundred years. There will be a copper shortage, by the way. So if you look at what we drag out of the ground, what we find is it's mostly fossil fuel and stuff we build from. And all the other stuff is orders of magnitude less. And so one has to keep this in mind. If you can get carbon-based conductors to replace copper, it will be a very, very big deal because we're up to our eyeballs in carbon. A few more things. I'm going to skip some things because I want to run out of time. I want to talk briefly about data centers. They're beginning to gobble up a lot of energy in the US and other parts of the world. And the projection is that, well, in the United States, today, it's about 3.5% of all the electricity used, predicted to go to 8.6% in the United States. The reason is, as you go to bigger and bigger large language models and other AI machine learning things, you're using an exorbitant amount of power. And Meta, for example, has a gigawatts center. The latest one is 2 gigawatts, and that's the number of flops. But that's just one data center of Meta. And so if you look at the big guys, it's really enormous. You compare it to an alternate technology. The little girl on the left uses 35 watts, can learn languages with orders and orders of magnitude less data, can tell the difference in cats and dogs, and all these other things. So we're missing a good architecture and also pattern recognition. So something to aspire to. The guy on the right is doing a physics experiment and can figure things out with, again, far less data. Instead of hundreds of megawatts, we're talking tens of watts and orders of magnitude less data. Let me just go a little bit on nuclear. Nuclear, let me just say, is very safe. It's 1,000 times safer than burning anything, coal, oil, gas, and it's not very dangerous. In fact, a third of all the radiation you're exposed to is in the food you eat, and it doesn't have any incidences. China has figured out how to build nuclear power plants on budget, on time. The green thing on the left, if the rest of the world can follow China, nuclear will be a viable alternative. And I'm not going to talk about fusion, except there's a neutron damage problem in fusion. And the neutron damage problem is that the neutrons displace the atoms. There's alpha decay, makes helium bubbles. The thing turns into Swiss cheese. And I'm trying to figure out how to actually get the material to anneal locally and get rid of the damage. If we can't do this, commercial fusion may not be possible because of the neutron damage. Finally, agricultural revolution. We've had the invention of fertilizer. We've had the invention of crossbreeding. And this has had a profound impact on the world. From 3 billion to 7.9 billion people, the amount of land put under cereal production did not go up at all, even though the population increased and the world is better fed. What's the problem? The problem is N2O. Fertilizer then is not picked up by the plant, turns into N2O. It's a very bad greenhouse gas. It lasts 100 years, and it's 300 times worse than carbon dioxide. As the world gets richer, we're going to use more fertilizer. This does not have a technical solution at the moment. There is a possibility that we can make microbes that enter into symbiotic relationships with plants. This was done by a company, Pivot Bio. I was on the board of a company that did the synthetic biology that did this. The company went bankrupt because it took 8 years to develop the microbes. So now in my lab, we're trying to do it by putting in many dozen genes at a time to figure out the technology so you can do combinatorial speed up in the development of microbes, not only for corn, but for all the major grains. If we don't solve the fertilizer problem, we have a real problem. Finally, we have a carbon dioxide problem. We'll need at least 10 or 20 gigatons to capture each year until we get rid of these hard-to-abate things. And just to give you the scale, it's an order of magnitude in weight of all the landfill. It's, in fact, on the same scale as the amount of fossil fuel we dig out of the ground, which makes sense. We're digging out fossil fuel at the tens of gigatons rate, and we're going to have to put down the remnants of the fossil fuel and carbon dioxide at the tens of gigaton rate. And so we do not have the technology to capture carbon dioxide from air. This is something I funded when I was Secretary of Energy. It costs $1 billion. It takes the carbon of a quarter of a large coal plant. Air capture is 5 times more expensive than need be. I'm doing something about that, trying to do something about that, and stumbled on a way that perhaps can decrease the energy costs and the CapEx costs significantly. The goal is $150 a ton for air capture, much less than $100 a ton for point source capture. If it works, it will be deployed. But right now, the technologies you have do not work. And so that's the problem. The final problem I want to leave you with is if you look at the United States, when we transition from wood to coal to oil, natural gas, and other transitions, it takes about 60 years. Now, what is the world doing? Every time there's a new source of energy, we just tack it on. And so, this revolution, it's a minimum of 60 years, but it could be longer, especially if the world doesn't get serious. There are many problems that we have to address. I hope that I've given the young scientists here a glimmer of what things to think about. I'm thinking of a half a dozen. And my excuse of not being a young scientist is everything I'm doing is all new in the last 8 years. So I wasn't doing any of this stuff 10 years ago. But with that, I'll stop. Thank you.
The topic is I want to introduce is called perovskite-based space photovoltaics, the future. So before that, I would like to briefly introduce our university. Our university called Westlake in Chinese, 西湖大学. So it's a very new university. This university is actually established in 2018. So we just 8 years old. And then you can see that this is our campus, not close to the real lake, but we still have our artificial lake in the middle. And then, so thanks, Professor Chu, for the nice introduction about renewable energy. I don't need to talk more about the importance of renewable energy, because you can see that the keep increasing of the non-fossil fuels was using power in the world, especially for the solar cells. The solar energy is receive more and more attentions. That is because this is kind of maybe one of the best ideal solution for the energy crisis. And then you can see that solar cells is relatively renewable and sustainable. So, however, for the single junction, still have the efficiency limit. This is calculated by the previous physics scientists, Shockley and Quasar, and they calculate that there's a theoretical limitation for single junction, which is 33%. So to solve this problem, we can use the tandem architecture, which means that we can use some of this part of the absorber to absorb this, for example, the blue light, visible light, and some of the absorber to absorb the infrared light to try to reduce the thermalization loss and increase the theoretical PCE of the tandem solar cells. And the perovskite, that's because and then we can use this so-called perovskite materials, which is relatively new materials was firstly designed or synthesized by Japanese scientists in 1929. And then this is ABX3 compound. A side could be organic cations, for example, organic ammonium, amidium, cesium, etc. B side could be the metal divalent cation, for example, lead and tin, and the X side could be the halide iones, for example, iodine, bromine, chlorine. And then this can have a very good combination with this traditional solar materials called CIGS, copper, indium, gallium selenide, and which is flexible. We can use these advantages of perovskite by the tunable dimensionality and the tunable band gap, and then also these advantages of 6 is also the band gap is tunable and you can make it flexible. So we can combine with these two advantages of the two materials together and get this flexible tandem solar cells. So why I mentioned this space is because that our land is actually limited, especially in China. So we have to make more applications for the solar cells in the satellite and the space, because space is actually limited. So we can take a look about this summaries of this mainstream photovoltaic technology. 6, you can see that it's not that high efficiency, but the price is just normal. But why I'm still working on the perovskite CIGS tandem is because when we apply this solar cell technology into the space, we have to care about the 2 parameters. The first one is the irradiation stability. As you can see that the 6 have a very good irradiation stability. You can see that when we make this tandem configuration, this always shows a slightly decrease after a significant amount of the irradiation, which equals to the maybe 5 years operation in the space. And then we can also see that the silicon have a significant decrease after irradiation. And the perovskite, some of the composition have a very good performance, and also some of these are still under test. So we are actually also working on this parameter. Another is that people care more about this power to mass ratio. And then when we make this CIGS, especially on the stainless steel or the PI film, you can see that the power to mass ratio is the highest. So when we make this tandem, so this have a significant advantages in the application of the space photovoltaics. And then we have these plans to launch this product because the gallium arsenide is still very perfect in the application. So when we try to compete with the gallium arsenide, we have to do very well in the lab, especially. So we are still in the phase 1. That is still the R&D. So what we are working on is try to increase the efficiency that can further improve this power to mass ratio. And then you can see that this the perovskite CIGS is actually, before our group get into this field, the efficiency is only 13%. And then after we get in, we can push the efficiency to 28%. And then right now we can make a over 30% perovskite CIGS tandem. So I will use the time to briefly introduce our research on the perovskite CIGS tandem. The first one that we want to introduce is the paper we published in Nature Photony in the last year. Try to using this divalent cation replacement strategy to try to stop the ion migration in the perovskite active layer. As you can see that when we incorporate this MDA and then the ions, especially for the iodine, it's really hard to move inside the perovskite lattice. And then we can improve the efficiency of the wide bandgap solar cell, improve from 22 to 23%, and then the stability also improve after several hundred hours operations, can still maintain the 80% of these initial efficiencies. And then we can make the tandem efficiency. Full terminal, we got a 30% efficiency, and then the flexible 2 terminal. This is actually the worst, the first so-called highly efficient 2-terminal perovskite CIGS tandem, which certified efficiency was 23%. And right now we move keep increasing this number to get this number of 28%. And then we also have some invention on this self-assembly monolayer, the whole transporting materials. We designed the first all-carbon pyrene-based whole transporting materials with improved stability, and also we incorporate this kind of orthogonal core, and then to introduce this amorphous whole transporting materials. And then by utilize this two technology, we can increase the efficiency of the large area perovskite CIGS from actually previously there's no report on the large area perovskite CIGS tandem, and then we can make the efficiency to 25%, which is comparable for the silicon single junction. And then it's also showing very good mechanical stability. After bending cycles of 10,000, it still maintains this 100% of the original efficiency. And then to facilitate the industrialization of this kind of materials, we also designed a new technology we called it to surprise the highlight segregation in the blade coating technology. And then we can print our solar cells by this slow die coater, and then the efficiency further increased to 27%. And then this paper was just published in Nature Energy last week. And thank you for this WLA to offer us this opportunity to present my work. Thank you.
It's really an immense pleasure to be here today and tell you a little bit about medical robotics. In my opinion, it's truly an exciting field, and actually maybe younger than we would think. So the first surgical robots were demonstrated about 40 years ago. About 20 years ago, then the first commercial systems entered clinics. And in the last decade, we really experienced a strong momentum with about now millions, about 1 million of procedures carried out annually. About the same, and for the robotic systems that you probably are familiar with, is such as the Da Vinci system that is now so heavily used. And about also in the past decade, we experienced also lots of momentum actually in the direction of untethered robots, such as this camera pill that can be navigated using magnetic fields through the gastrointestinal tract for endoscopic interventions. And during this time, so pushing this idea further, in the robotics community, we've been thinking about also scaling this further and really creating wireless controllable medical micro and nano robots that one day could move from within the body to deliver drugs more effectively. Indeed, until today, the inefficacy of drug delivery remains one of the major hurdles in medicine and one of the key reasons for the failure of phase 3 clinical trials. More than a century ago, Paul Ehrlich already postulated the idea of a magic bullet. He said we need to learn how to aim chemically. And we made tremendous progress in this short distance targeting and how we can actually biochemically target, right? But I think we still have to overcome large physiological barriers to really do this long distance and move drug carriers more effectively from an injection site to a target site, such as tumors. Now, what you see here is a millimeter-sized magnet, about 300 micrometers in width, that is moved in silicon oil with 5 degrees of freedom control, so free translation to rotation. So how about we would just apply this concept and move now magnetic nanocarriers filled with drugs to a tumorous site? Well, unfortunately, physics is not on our side here. The force scales with volume, which is negligible at the scale, and also with the gradient, which decays rapidly about the inverse of the power of 4 with distance. And on top of that, we're entering the low Reynolds number regime, where viscous drag forces are dominating and inertial forces are essentially not important. So, as so often though, nature might have a solution. What you see here is actually a bacterium that is chased by a macrophage, and it moves very effectively. So if we take a closer look here, what you can see are bacteria that swim, and they have cilia or flagella that are spun by a molecular motor, executing this non-reciprocal motion, translating them forward, which is very effective even in very highly viscous environments. And so this inspired really the robotics community, and because of which most of the first micro robots were actually bio-inspired, trying to mimic this very effective propulsion. So here's an example of such an artificial bacteria flagella that can be actually 3D printed by a two-photon photolithography. We can then batch fabricate, coat them with magnetic materials, and then apply now. So the molecular motor is now a rotational magnetic field that we can very effectively transmit also over wide distances that is spinning these helices that can then be controlled. And actually the first clinical microrobots also were just commercialized for neurovascular applications. But now if we really want to move out of the vasculature and go deep into tissue and target, for example, tumors, we need to get smaller and we need to be many, or we need to have many. And so why not actually using real bacteria themselves and not trying to mimic them if nature provides already such great solutions? And in a way, if we can externally control them, they somewhat behave like, or they somewhat are smart microrobots. They have onboard sensing, they can follow chemotactic gradients, they can also produce actually functional molecules that we want to potentially could use for therapeutic applications, and they of course self-propel. So we can, so this gave rise to the field of biohybrid microrobots, where we use living cells such as bacteria or also other cells, and then we augment them with functional nanomaterials to basically give control or also expand functionality. So now to leverage the magnetic control, we need to be able to move them magnetically, right? And so luckily nature has a solution for that too. There do exist magnetically responsive bacteria that are also referred to magnetotactic bacteria. It's an aquatic species that biomineralizes these beautiful iron oxide nanocrystals, mainly magnetite and greigite, and they use this to navigate with the Earth magnetic field. Now when we superimpose a directing magnetic field, we can guide their direction, right? But what's even more powerful, and which doesn't rely on their molecular motor, is when we apply, again, rotational magnetic fields that we really can transmit well over distances. So you see here red fluorescent tracer nanoparticles mixed with a swarm of these magnetotactic bacteria, and when we generate rotational magnetic fields, we can provide these large volumetric flows through ferrohydrodynamic coupling. And we studied this quite in detail, don't have the time now to go into, but we were very encouraged by this approach, and so were others. So what we started to do was then actually covalently coupling now the nano drug carriers filled with drugs such as chemotoxin 5-FU onto the bacterial membrane using copper-free click chemistry. So we have this really drug delivery vehicle, living drug delivery vehicle. So how can we now get out of the vessels, right? This is the idea, and to penetrate into tumor tissue. So to overcome the elastic resistance, if you need to squeeze through endothelial cells, which we modeled here, the molecular thrust of a bacterium would not be high enough. So, but what also happens actually when we, in this top-down view on an endothelial barriers, actually it's a stochastic process. These gaps open and close. And so when we now apply these rotational magnetic fields, what happens is that these bacterial vehicles can actually roll along the vessel walls and add increased surface exploration and the chance to find a gap to basically move through, which is more likely than if you're just trying to navigate straight out of the vessel and rely on the molecular thrust. And so we could match actually, or we could find actually similar experimental validation than to also to our in silico model. So next, as we exited the vessels, we want to penetrate tumor tissue. Again, we continuously apply these rotational magnetic fields. You see here a tumor rim, so this is a 3D tumor spheroid model with a human breast cancer cell line. Here you see in red labeled our biohybrid microrobots that could very effectively colonize this tumor. And the beautiful approach about this is it's a hybrid approach. So we use first the external magnetic fields to help and craft and get out of vessels into the tumor. But then when we switch off the field, they still have their chemotactic gradients and actually seek the hypoxic niches of tumors. And we could also demonstrate this then in vivo trials that we could significantly increase the number of drug delivery vehicles at the tumor site. So if we when we want to do this, we want to activate this approach only at the tumor site, right, and nowhere else. So we need to spatially focus our rotational magnetic fields. And we do this by actually borrowing principles from magnetic particle imaging. So we use permanent magnets to generate a zero field point. Then we superimpose a global rotational magnetic field that now only freely rotates in the zero field point and is suppressed in these off-target regions outside. This is how such a system design looks like for a mouse scale setup with an AC coil for global rotation, a cut magic sphere 3D Hallbach array to generate the zero field region in 3D, and then AC coils to push that zero field point that we can actually pinpoint and target. This is how the system looks in real, and we're working also on new designs that are actually more accessible for clinical applications. Now, with the rise of living therapeutics, we're really excited to transfer this, in my opinion, powerful approach also to other cell types, not just these magnetotactic bacteria. So we started to magnetically functionalize genetically modified E. coli Nissl bacteria with therapeutic function. They produce certain toxins. You see in brown, we false colored the iron oxide nanoparticles, how we decorated the E. coli Nissl, colored in blue. And you can see that we can also apply now these rotational magnetic fields. All this is actually only possible through the convergence of science, and I feel we're really at an exciting point where biology and robotics are merging, and we can basically come up with completely new tailored biomedical applications. With that, I would like to thank you for your attention, and to my whole team, with whom I feel very privileged to work with over the years. Thank you very much.
Okay. Unfortunately, these are not the slides I sent, but the talk I was going to give was about the molecular mechanism of, or our current understanding of the physical basis of chromosome condensation. So I'll do this without the slides. I was going to begin by presenting a common reference point, which is the process of mitosis, or somatic cell division in eukaryotes. And I was going to show you representative stages of the process of mitosis as sketched by its discoverer, Walter Fleming, in the late 19th century. And it would have begun with a hand drawing of an interphase nucleus throughout which the genetic material, or chromatin, is dispersed. And what is seen in Fleming's drawings is that the genetic material, the chromatin undergoes a process of what I call chromosome individualization, whereby the genetic material converts from its dispersed state to individual chromosomes. This occurs during prophase and is complete by prometaphase. Thereafter, the chromosomes congress at the metaphase plate, where they form attachments to spindle fibers emanating from the spindle apparatus, which pulls apart, at anaphase, sister chromatids disjoin and are pulled to opposite poles of the cell, where in telophase, the chromosomes undergo essentially the reverse process of decondensation, thereby reconstituting the interphase nuclei of the daughter cells. And this process that I've called chromosome individualization is more properly referred to as metaphase chromosome formation. It can be conceptually decomposed into three events. First being the formation of a linear axis. The second being the emanation of loops of chromatin from that linear axis, which we've learned in the past 10 years or so is driven by SMC proteins like the condensins and cohesin. And this process of loop extrusion brings about linear contraction of the chromosomes. And the third aspect of metaphase chromosome formation is volumetric compaction of the chromosomes, or chromosome condensation. And that aspect alone will be the remainder of our discussion. I was then going to show the relative change in volume of the chromosomes. It's between the condensed state in metaphase and the decondensed state in interphase. It's about 2 to 3-fold. This has been measured by For instance, three-dimensional fluorescence microscopy, where one can follow a cell as it enters mitosis and as it exits mitosis. And for a long period of time, the conventional explanation for this effect was that the chromatin was interconverting between 10 nanometer fibers and 30 nanometer fibers. This idea dominated chromatin science for some 40 years and was, it originated from a landmark study by Aaron Klug and colleagues at the LMB, which was published in 1976 on the salt-dependent compaction of chromatin fibers in vitro. And while it is, there's no doubt that chromatin can form well-ordered 30 nanometer fibers in vitro, and this is a dominant mode of organization in certain specialized cell types like avian erythrocytes. It is increasingly doubt has been cast on the generality of the idea, and with the evaporation of support for the 30 nanometer hypothesis as the basis for condensation, we therefore cast about for alternative explanations. And to do this, we required a means of measuring chromosome condensation in vitro in a manner that allows us to vary the condensation state by applying arbitrary environmental perturbations. And no suitable methods were available at the time we began this work, and that's for a very simple reason, which is that chromosomes are exquisitely sensitive to damage. I was then going to show figures from a 1997 paper by Hoekmanzadeh et al. John Marker was one of the authors, and he's a notable person in chromosome micromechanics. That paper shows a force-calibrated micropipette, or the figure I was going to show shows a force-calibrated micropipette latching on to a chromosome from a bundle of, from a chromosome cluster that they liberated from a cell. And these authors found that chromosomes respond elastically in the range of about 1 nanonewton, but they undergo irreversible deformation when subjected to forces of the order of 10 nanonewtons. And in a very common, or what has been a very common method of preparation of samples for microscopic observation, one begins with chromosomes in suspension, deposits a droplet on a glass slide, and then seats a cover slip. But in so doing, the meniscus of the mounting medium is set into motion, and the advancing contact line can generate capillary and viscous forces of the order of micro to millinewtons. And what one finds if one's prepared chromosomes by this method for any considerable length of time is that there are a great many chromosomes destroyed. And common practice is just to ignore those and focus one's attention on on those chromosomes that appear to have survived the procedure intact. But we thought it better to bypass this mode of damage altogether, and so we adapted a parallel plate flow cell, which we can preassemble and into which we can inject the suspension of chromosomes. And we can functionalize the surface of that chamber in a suitable manner that is conducive to the capture of chromosomes and their gentle immobilization in a manner that doesn't restrict their subsequent expansion and compaction. And we can then, this is optically coupled to a high numerical aperture objective lens, and we can freely exchange the solution that bathes the chromosome by means of an attached pump. And by doing that, we can vary the environment of the chromosomes and image the changes in their condensation state. And I was going to show images of the chromosome in decondensed and condensed state. To make these observations quantitative, we then developed procedures for the distillation of morphological descriptors from the image series that we collect. So we can determine things like the length of the chromosomes, the width, the orientation, the area. And we do this with classical computer vision procedures, which draw heavily on from the branch of mathematics known as morphology, so erosion and dilation operators and so on. And we can then summarize the results in the form of a condensation profile where we normalize the degree of condensation relative to two calibrants that we include in every experiment that prepare the chromosomes in a highly decondensed and a highly condensed state. And then we plot this degree of condensation as a function of some perturbation. For instance, the concentration of spermidine, which is a well-established physiological regulator of chromosome condensation. And we can conduct this analysis at the level of individual chromosomes because we obtain a condensation profile trace for each individual chromosome, or we can average them and see the bulk behavior. And well, we can very precisely tune the degree of condensation. And importantly, the process of chromosome condensation is reversible. This slide is in this slide deck here, so I can actually point to this one. So here we see chromosomes in odd-numbered cycles of an experimental sequence in which spermidine was alternately provided and then removed. And so the chromosomes convert from a condensed state to a decondensed state and back. And we can determine the degree of condensation. We can also show that the morphology of the chromosome is unchanged by Pearson correlation coefficient analysis on the images. And then this can be carried out indefinitely, limited only by the patience of the experimentalist, which in my case was 10 cycles. I then go on to show that that chromosome condensation, decondensation is isotropic. So we have the same change in length, width, and height. And I showed an example of a chromosome in a condensed state and a decondensed state. The difference in volume is a factor of 10, roughly, and then the difference in each linear dimension is a cube root of 10 or something, 2.3. And we can show this by three-dimensional analysis, where we take one of the chromosomes and in silico expand it, or in silico compact it and superimposes very nicely, and get a very high Pearson correlation coefficient between that and the experimental image. We can also show this by constancy of the aspect ratio of chromosomes in different condensation states. And so then, equipped with this system, we proceeded to undertake a systematic exploration of physicochemical parameter space with the aim of identifying key determinants of chromosome condensation behavior in vitro. And I am only able to summarize the conclusions for lack of time. I began by showing a series of condensation profiles that are produced by a family of linear amine compounds, the polyamines, varying in nominal charge from +1 to +4. And we would have seen these spaced apart in a manner that shows the exponential dependence of condensation on valence. And I then show good agreement between theoretical expectations and the experimental results. In particular, we turn to a well-established thermodynamic theory of polyelectrolyte gel swelling, which was developed by Paul Flory and John Renner in 1943, in which the free energy of the gel admits a decomposition into three terms: that of polymer-solvent mixing, rubber-like polymer elasticity, and the entropic penalty of counterion confinement, which we model according to a Gibbs-Donen potential. And the system of equations cannot be solved algebraically except in the simplest of cases, but we're able to produce a numerical solution which shows good agreement with our experimental observations. And we concluded according to this that chromosomes are polyelectrolyte gels. I then went on to show that condensation is not monotonic as a function of salt, and that in fact above a certain threshold condensation, above a certain threshold ionic strength, chromosomes reentrantly decondense, and this kind of reentrant behavior is a characteristic feature of polyelectrolyte gels. And I then showed a paradoxical result, which is that the condensation is not only non-monotonic in the ascending direction of ionic strength, it's also non-monotonic in the descending direction. And so when one drops the ionic strength to very low levels, about 30 micromolar, the chromosomes paradoxically condense. So our conclusion then was that Well, that was an expected feature of polyelectrolyte gel swelling, despite its paradoxical nature. We conclude that chromosomes condense and decondense according to a volume phase transition. I was going to describe a theoretical work by Carl Duszek and Donald Patterson published in 1968, which was verified experimentally by Toyoichi Tanaka for polyisoproyl acrylamide gels in 1978. And basically, just about 30 seconds, this is called a volume phase transition. It converts between a dense gel and a rare gel state, and this explains the condensation behavior of chromosomes. So our conclusions are that chromosomes are a two-state polyelectrolyte gel, and their condensation-decondensation behavior is mediated by a volume phase transition.
And the project is known as Quest DMC. This was originally imagined as a twofold enterprise. The first one being about using helium-3 as a target for detecting dark matter, or at least probing a certain part of the parameter space where we believe there might be dark matter. The second one is about taking one of the rare, maybe even the only 3D system, quantum system with a first-order phase transition that we can isolate from any external influence, and trying to understand how that phase transition works. And then hoping that this might tell us something about what happened in the early universe, provided some of the ways that have been speculatively used for expanding our current understanding of the standard model of particle physics and so on, are actually correct and describe our universe. Later on, it then turned out that that actually these, technically speaking, are more or less the same project. Both of them are kind of building the basis for the other one, and that's been very entertaining to kind of learn during the process. Now, dark matter, what is that? Well, let's say if we follow how stars fly around the centers of a given galaxy, we measure the velocity, and then we compare that with the amount of gravity that we can account for. The gravity available does not seem sufficient for holding the stars on their trajectories at the observed velocities. And to explain this discrepancy, then, a common hypothesis is that perhaps there is some form of matter producing gravity such that we can't see it. And that's called dark matter. There is no direct observational evidence that it exists other than the gravity effects of various kinds, including this particular observation. And so therefore it's been a challenge for a long time already to come up with an experiment that can actually observe some sort of direct interactions locally with this potentially existing matter. Now, we don't really know very much about this matter, even if it does exist. For example, we don't know whether it's formed of a single particle, and if so, what the mass of that particle is. And the range of possibilities is rather large. So it ranges from light enough so that the de Broglie wavelength of the particle is of the scale of the galaxies, the structures that we know that this thing interacts with. That gives you very light particles. And then if you allow the mass to be very large, well, then there are some fundamental limitations from well, I don't know, black hole formation or some other dramatic phenomenon like that. But this leaves like tens of orders of magnitude of range. And traditionally, people have looked in two different places, motivated by some independent theoretical speculations or other reasons. The blue circle is the wave-like stuff, for example, axions, have not been observed. People have tried. It's not very easy. The red circle is well, mostly so-called WIMPs, weakly interacting massive particles. And these, well, they arise from some expansions of the standard model, but they have not been observed either. And so what people have recently started doing is, well, basically looking everywhere where they can instead of just in some particular places, and this is exactly what we are doing. We're looking in the green circle, which is basically particle-like, but lighter than the traditional WIMP range, and also lighter than for scale, for example, than a single proton in most cases. This image is stolen from the Quantum magazine. Now, helium-3 is a good target for looking at this region for the reason that it's light, but and the state that it forms has a spin, so it can interact in ways that require that. But then the kind of most important reason is that it forms an exotic quantum state where we can, it turns out, measure imposed energy from a collision very precisely. And while I will skip most of the reasons why that is the case, I will just say that it's a fermion. That means that to form a superfluid or any macroscopic quantum state, we need to first pair the fermions. And because these atoms have a finite size, this is a complicated task. And then we have a lot of complexity, and we can choose a region in the complex behavior of the system which is not particularly sensitive to any added energy. That's the front corner of this phase diagram at low pressures and temperatures. Now, the experiment itself in the computer-rendered image is on the right, and essentially what we do is we take a small amount of this superfluid. So you may have heard of xenon detectors, for example, that weigh several tons. This one weighs 0.1 grams, so that's a stark difference. And well, obviously we need to use very low radioactivity materials and be able to measure energies imposed in the system down to very small values. That's achieved mainly by cooling the system down to an extremely low temperature, around 100 microkelvin, so 100 millionths of a degree above absolute zero. And then basically we move some mechanical object in there, and any temperature, any thermal excitations in this system are observed as a mechanical drag force on the mechanical probe, and that's how we know what's going on there. The target itself is kept in a little box. These are some prototypes. They're not the final things, but they're nice because these are made from glued millimeter paper, so you can really see the scale. The little holes are for thermalization, and then some mechanical probes you may be able to see inside of there, vibrating little objects. And this is the temperature of the superfluid inside of the box, measured over a period of time in a prototype detector. And the peaks which you see are background events from cosmic rays and maybe also some radioactive processes taking place in the materials in the room. And then basically the task here is to improve the sensitivity. So here we can measure about a kilo electron volt, and we're going to need about 10 electron volts. This was measured 2.5 years ago, and at that time we'd basically reached the state of the art that anybody else had done. And as of last week, we started running the a proper detector, and we have now tentatively reached about 10 electron volts sensitivity, which is 2 orders of magnitude better than anybody had done before. And so thus we are entering this white region, which you can see in this piece of the mass interaction likelihood parameter space plot. And so as soon as we've been running this for a long enough time, and also obviously understand the backgrounds and various other things we haven't done yet in this energy range, we'll be able to exclude that. And so then kind of bring our little contribution to this quest to understand, well, in this case, probably just like all other dark matter experiments, where dark matter is not. I'm not saying we are very likely to find it. And I think I've used most of my time here, so I will just thank you for your attention.
So what is my problem? My problem is about deformation. When I was born like a small boy like this, a small kid. So I grow up. I deform until I become what I am today. And I'm pretty sure that one day I'm going to die. When I will die, I will decay again, deform, and go back maybe to 2D or 1 dimension. And another problem is when I have a balloon, if I pop the balloon, what happens is that air goes outside. And that balloon deform until it stops being a spherical figure. It becomes now something that is flat. We also see it in the movies where they were speaking about transfer a robot that changed. It became liquid after it became again a solid. And I asked myself the following question. If I was an element or an object, am I an object of Euclidean geometry? And I told myself, I'm not too sure. So in mathematics, of course, we have what we call a complex number. We have what we call real number. Complex number is 2 dimensions. We have quaternion that is 4 dimensions. Then we have octonium. So mathematicians like to go to higher dimension, sometimes lower dimension, to describe the world within which we stay. Whereas we are in 3 dimensions. So I decided to, to introduce a new concept, not new concept, just a modification. Of course, that certain mathematician will not be happy with me because mathematicians like what they call associativity, they like what they call commutativity. If you are not commutative, sometimes they are not sure. Whereas when I look at the nature, I notice that nature is not commutative. So it means that somewhere the idea of confining mathematical geometry in terms of commutativity may be a problem. So you see that as I was speaking, real number, complex number, quaternion, octonium. Yes, they are powerful, elegant, mathematically complex, but the question is where it is then this 3-dimensional algebraic structure that we can use, So I started something. I decided to go in 3 dimensions. Of course, the first formula is not new. You can see it in the literature. So I have what we call A square. It is imaginary. It's equal to -1. G square also equal to -1. Now I want to introduce into my formula a deformation. Of course, for this deformation, I must make sure that I dance between commutativity, non-commutativity. So what I do now, I will multiply I and G. I will say is equal to 1 minus alpha plus alpha K. And I will say Gi is equal to 1 minus alpha plus minus alpha K. Then I'm going to introduce this information here. Ig, Gi, Ig is equal to 1. So my R at the moment is going to be a real number. But if I find now K in terms of Ig, I can now restrict and say, okay, my alpha will be different from 0. So this is the setting. I have not presented here, for example, the multiplication. So I'm going to present to you the multiplication. So now, of course, all of us will know about a complex number. When you say I square, my I square is equal to -1 is what they taught me in school. But I'm not too sure that that is what nature is. So what I did is that with what I introduced before, my K square is equal to 1 minus 2 divided by alpha. So let me tell you the reason of this. I would like to have a mathematical structure that can explain what they told me in the village. My younger brother told me he was going in one village to date a lady, and he saw a man that decreased in size and the person disappeared. And I'm not sure whether the story is true, but the imagination that the person disappeared, how do you use mathematics to explain the fact that somebody can disappear? So if you see here, if I have, this one is the 4th dimension, and if you take alpha equal to 2, for example, you're going to see that my K square is equal to 0. It means that I have lost a dimension. I'm not nilpotent. So now I present to you my multiplication table. This is my multiplication table for this structure. And of course, if alpha is equal to 0, I have commutativity because I is equal to minus, G is equal to minus I. If alpha is equal to 1, I have quaternium. So now if alpha is equal to 2, I have emergence of 3-dimensional nilpotent regime. But now if I'm between 0 and 2, I have something very interesting. So now I say let me redefine what they call distance, because in Euclidean geometry, distance is x squared plus y squared plus c squared. But now, if I take my z and I take my my conjugate, so I put them together, that's what I obtain here. So I have a distance that is able to show me flexibility. I can press, I can enlarge. So this is what I obtain. But if alpha is equal to 1, I obtain the geometry that they taught me in high school. Oh, so now I can define a circle, I can define a sphere, I can define also what they call cylinder. So this become non-rigid, it become flexible, it can be manipulated, it can change. So this, when you introduce something like this in mathematics, there are many fields that have to be reformulated. For example, fractal. In my country where I come from, fractal, there are something, for example, like what we see in my code like this. But the Mbange brought one that was introduced. It's not reflecting this. So now I can use this concept to introduce new kind of fractal. I'm going to show you the video later. So in fractal, very simple formula, this is what I obtained. And so I will show the video a little bit later. So now I want also to trigonometry. I want to define what they call trigonometry. This is in 2 dimensions. Normally we are supposed to be in 3 dimensions. I cannot understand why they are telling me about angle in 2 dimensions, whereas when I look at you, I see you in 3 dimensions. So is it possible for us to have angle in 3 dimensions, forget angle in 2 dimensions? So I redefine what they call trigonometry, for example, cosine, sine, and a tangent. So I get my own renaming to rename it. So now, I move to another section, partial differential equation. Of course, people were speaking about diffusion, wave. Now, I want to redefine what I call a partial differential equation. So from now on, if you are using diffusion, you can speak about anisotropy. So wave equation, for example, so this is how I redefine in a simple way my Laplacian. And also people that are working in physics, they can use this idea to redefine maybe Einstein equation, to redefine space-time equation, and everything that we want to redefine as a mathematical. So I'm going to conclude here for presentation because I know some people don't like many formula. So now I'm going to show you the figures of what I have presented here. Okay, so for example, this is my Trinitron cylinder. I can see now it is flexible, it can breathe. My Trinitron cylinder. Here, it is what I call fractal. So I don't know why it's not working. Normally, it is a video. It shows you how this fractal change. It change, and it becomes something else, as you can see, how it's working here. So this is the first time, perhaps I'm not too sure, but I think that is the first time ever in mathematics we are able to see a fractal, if I can, I'm not saying being, but something that it changed the form according to the deformation parameter. So you will see when it's going to pass 0, it's going to change the form and you can obtain something different. Okay, so since the time is going, I wanted to show another one. So you can see that anytime that alpha changed the value, we have a new type of fractal, we have a new figure that gives something different than the one that we had before. Okay, so this is another one. This is in 3 dimensions. I want to show something different. So this is, I call it breathing cylinder because it can elongate alone, it can compress alone. So this is a mathematical formula that is able to express, to show us this type of simulation. So I want to show you, I call it African wizard sphere. Why? Because it can appear, it disappears. This is a mathematical formula. This is what I want to see. This is another one that shows me how something generates and after it degenerates. My idea was to introduce a 3D complex system that is able to take into account deformation. So since the time is finished, thank you very much. This is what I wanted to show you.
So I'm going to talk about embodied intelligence. So once intelligence meant something very simple. It meant that a living system could survive in a world that resisted it. An animal was intelligent because it could adapt and act. A human was intelligent because experience changed them. Today, we use the same word for machines, for systems that can write, reason, and drive. We call them intelligent too. But by doing so, we have quietly grouped together things that experience the world in very different ways. So in a sense, there has been a silent redefinition of intelligence. For biological systems, intelligence emerged from consequence. They act, the world responds, and they change. For machines, intelligence is now defined by performance and scale, by how well a system can model data. Well, what's happening right now? Without noticing, we shifted from learning through experience to learning through labels and observations. Now, there is a spectrum in how systems experience the world. Language models learn indirectly from records of what others have done. Autonomous systems are a little better. They act in the world, but again, with fixed goals. Biological systems learn from their own actions directly and open-ended. The difference is not merely capability, it is consequence. Models trained on data learn what usually happens, but they do not learn what happens when they do something new. They can see the world, but they do not change it. And without change, without tracking that change, without modeling that change, there is no consequence. So embodiment is not merely about having a body, it is about completing the loop between perception, action, and feedback so that the world can push back. That push is what gives experience meaning. For biological systems, this loop actually has structure. They perceive, they receive feedback, they act, they learn from it, and very crucially, they discover new affordances. It is the closed coupling of these five that makes intelligence embodied. And as I said before, autonomous systems do move closer. They perceive and act, but their goals are fixed, and their learning is narrow, and their affordances are predefined, all things which we want to go beyond. So they execute within a world, but they do not yet discover it. To the best of my knowledge, we don't have autonomous cars going out from their parking lots just to see what the world outside is like and what would happen if they did drive a little bit differently, go off track. So this reveals the real frontier. So intelligence is not defined by what a system can do today, but how it changes through interaction. Perhaps the next generation of intelligence will not be something that we train again, and deploy. Perhaps it will be something that grows slowly, unevenly, and irreversibly through embodied experience. So we are not just talking about coming up with a new architecture. It is a new rethinking, a new definition of intelligence that we want to go for. There is a line from ancient Indian thought which says, Seriram Kalu Dharma Sadhanam. So that's in Sanskrit. And translated, it means that the body is the instrument of purposeful action. It reminds us that intelligence is not just computation, but engagement with the world and consequences. So our challenge today is to bring this to life in the systems that we build. Thank you so much.
I would like to begin with quantum computing. See, existing crypto systems use classical bits, while quantum computers will use qubits. A single classical bit can be either 0 or 1. So there are only two possible states. If we take 2 classical bits, then we have 4 possible states. And if we take n classical bits, we have 2 to the power n possible states. But this is not the case with qubits, which are used by quantum systems. A single qubit is either 0 or 1 or both at the same time, and this is called superposition, which means many states at the same time. Here, the independent states act as the basis states. That means if we have n qubits, there are 2 to the power n basis states, and there are linear combinations of these 2 to the power n basis states over 2 to the power n dimensional quantum state space. And this is what we call exponential scaling. The exponential increase in the size of the quantum state space that gives the core strength to the quantum computing. Apart from this, there is another property that is exhibited by qubits, that they can be linked together. So the state of one qubit depends on the state of the other, and changing in one bit can affect the other even if they are far apart. This is what we call entanglement, allowing the quantum computers to process information collectively and explore solutions faster. Quantum computing also utilizes the quantum interference to rule out the wrong answers and amplify the correct solutions with the principles of destructive interference and constructive interference. And this is how these properties allows the quantum computing to challenge the existing crypto systems, which safeguards our financial transactions, medical records, government communications, and secure internet connections and whatnot. The risk to today's crypto systems is not sudden collapse, but delayed exposure. Data encrypted today is currently unreadable. It can be stolen and stored awaiting decryption technology that will be capable making it readable in future. This is what we call harvest now and decrypt later. The emergence of quantum computing forces us to re-evaluate and reset the assumptions for digital security. Here goes the Shor's algorithm, and see what it does. This is Shor's algorithm is when we run it on a potential quantum computer, can expose the secret key of RSA crypto system by factoring large numbers or break the discrete logarithm problem behind elliptic curve crypto systems, Diffie-Hellman crypto systems, or algorithmic crypto systems. And it can factor a 2048-bit RSA key within few minutes. So these popular crypto systems could be completely broken by quantum computers. We have Grover's algorithm that will run on quantum computers, will speed up guessing secret keys and accelerate hash attacks by reducing the difficulty from 2 to the power n to 2 to the power n by 2. That is, it halves the exponent. Means it can search an unsorted database of size n in n steps if we take classical search, but with quantum search, it will take under root n steps. That means if size of the database is 100, then quantum search will take just 10 steps, and classical search will take 100 steps. And that's why the security of AES 128 gets reduces to the security of 64. So the preferred choice is to double the key size, and that's why we use AES 256 these days. So in the post-quantum era, we need potential public key techniques that can withstand quantum threats. But we need not abandon the symmetric key cryptography, but we need to strengthen it with the use of larger key sizes. So what we need to do is that we need to adopt post-quantum cryptography, which deals with the encryption techniques that will stand the attacks posed by quantum computers. So here, the approaches will not rely on number-theoretic problems, but they will rely on the new problems like lattices, codes, and hash properties. Here goes some of the promising PQC approaches, and the most promising one is the first one, that is lattice-based cryptography, which hides the secrets in high-dimensional geometry where finding structure is provably hard, even for quantum computers. Lattice-based schemes relies on very hard problems like shortest vector problem, closest vector problem, learning with error, ring learning with error, and module learning with error. And the module learning with error is the recent one which balances the efficiency like ring learning with error, and which also provides the conservative security like learning with error. The next candidate is hash-based digital signatures, which differ from its counterpart, which is classical hash-based digital signature due to its threat model, construction goals, and standardization maturity. The first approach, lattice-based cryptography, is a much preferred approach because it also allows some advanced structures like digital signature, key exchange, and fully homomorphic encryption that allows the server to carry the computations over the encrypted data. The last two approaches are not much harnessed because the code-based signatures or encryption suffers from large key size drawback, and multivariate quadratic cryptography also vulnerable to weaker parameter choices. So there are certain NIST PQC standards that have been recently published. So first two and last one, standard 203, 204, and 206, these are lattice-based, based on lattice-based algorithms. And the third one is stateless hash-based digital signature algorithm. The first one is based on the recent version of learning with error, that is module lattice-based key encapsulation mechanism, and it is the primary standard for key exchange and general encryption purpose. The second one, MLDSA, is also based on module learning, and that digital signature that is at the end 206, that is fast Fourier over NTRU lattice-based digital signature algorithm that serves as the backup to the MLDSA in case its lattice-based mathematics gets compromised. And the benefit of a stateless hash is that the digital signature scheme does not require the signer to maintain or update any secret between signatures, thereby eliminating the risk of state reuse attacks. So now I come to the main point where I believe the next decades of cryptographic research must concentrate. Here are certain key challenges. The first one is reducing key size and signatures size, because post-quantum hardness is achieved through high-dimensional structures, and shrinking them directly undermines the security against quantum attacks. Second challenge is tightening worst case to average case reductions, that is about making sure that mathematical problems used in cryptographic systems are almost as hard to break as the hardest lattice problems known in theory. Right now, the connection between two is weak, so we must choose parameters carefully to strengthen this connection to achieve better security. Then comes side channel attack. But does that not break underlying mathematics? Instead, it leaks secrets through physical behavior of the devices like timings, operations, power consumptions, electromagnetic radiations, and these aspects are not covered by mathematical proofs. So it's a big challenge, and it requires us to devise new quantum models, which exists as the open problem. Next one is that we do have standardized models by NIST, but standardization doesn't mean proven security. We recall that SHA-1 and even RSA variants were standardized long before serious weaknesses were found. Then better bounds on quantum speeds are open challenges because we do not exactly know how much faster quantum computers can solve the hard problems, as we have no clear limits. And that's why cryptographers should not be overestimating the available PQC approaches, and assuming the worst cases, we should take larger and safer parameters. For the future directions, we need low-powered PQC because undoubtedly PQC algorithms are computationally heavier in real-life applications, are full of low-power devices, as we have sensors, smart meters, wearables, and medical implants. We also require secure key storage as a future direction because post-quantum schemes use larger and more leakage-sensitive and long-lived keys that exceed the design assumptions of existing secure storage mechanisms. Side-channel secure implementations are required because post-quantums are highly sensitive to physical leakage and practical security requires new definitions. Now, it is time that when we need collaborative efforts, we need mathematics to build hard problems, computers, science to implement them, and policy experts to set timelines and standards. Because cybersecurity is a global opportunity, and it's our responsibility. Thank you so much.
Today I'm going to be talking about cancer and some of the research I did looking at how cancer spreads throughout the body and some interesting findings that we have. So I won't talk too much about cancer in the background. We're all familiar with cancer. We know about it. We have someone who's either been affected by it or impacted in our lives. But what you might not know is that cancer is most deadliest when it spreads. So say you have a single tumor in just your brain or your lung or breast, that's not what kills people. It's when cancer spreads throughout the body that causes a majority of cancer mortality or cancer deaths. So this happens through a process known as metastasis. So this is a process where you have a primary tumor, say in the lung, it enters the bloodstream, and then it travels through the circulation, and then it exits the blood vessels at, say, the kidneys, a different side of the bladder, brain, somewhere else, and it begins to form more tumors, and these are called secondary tumors. So what I did is we looked at a specific system of how these cancer cells, once they earned the circulation, how they were exiting. So the prevailing theory or the prevailing hypothesis is that tumor cells use a method known as diapedesis, which is common for white blood cells and immune cells, where they squeeze through the endothelial cells of the blood vessels, similar to how white blood cells squeeze through when they're chasing or having some type of pathogenic reaction. And so cancer cells are known to be able to use this method as well. But what we discovered is that cancer cells, in addition to this method, can also use another method to exit cells or to exit blood vessels, a method that we termed angiopolosis. And this is much different because the vasculature or the cell walls actively restructure themselves to push out the cell rather than it squeezing through. And this was a previously undiscovered or unknown way that the cells could exit. And so this was interesting because we also found that circulating tumor cell clusters, or groups of 2 or 3 or more cancer cells that are traveling as a group, can also use this method to exit the blood vessels. So it was previously thought that the circulating tumor cells had to first break apart or disassociate into single cells and then squeeze through one by one. As you can imagine, a big group of cells can't all squeeze through at the same time. So it was thought that they had to break apart, but what we saw, or what we found out, is that these circulating tumor cell clusters could actually utilize this method, angiopolosis, to exit the blood vessels as a group while maintaining their multicellular phenotype. And so why this is important? So the clusters are especially important for us to study and to understand because, single cells, they often remain dormant. So if you just have one tumor cell or one cancer cell, that's usually a lot less dangerous than if you have a cluster or a group. It's the clusters and the groups that are really the ones that are more dangerous. They have more metastatic potential, more ability to spread and survive at different sites. So these are the ones that are causing a majority of all cancer and cancer spread. And so it happens through a multi-step process, but the real-world analogy I like to use is that cancer cells, when they travel as a group, it's analogous or comparable to a foreigner traveling to a distant place or a foreign land. And instead of you traveling by yourself, you bring your family, you bring your friends, you bring your colleagues, you bring people that you're familiar with, and you'd have a much better chance of being successful in that new environment versus if it was just you by yourself. So cancer, they do a similar thing where when they're in a group and when they bring their own structure and support, they have a much higher chance of forming these secondary tumors in a distant site because it's actually a very highly inefficient process, metastasis or spreading. Because if you think about it, in the example, you have a lung cancer and it's traveling to a completely different site, say, a kidney or brain, that's a completely different organ system. It's a completely different microenvironment that the cell is used to. So although a small percentage of cells actually successfully spread to a new site, they account for the majority of all cancer-related deaths. So what we did, so I told you kind of what we found out, but I'm going to tell you now how we found this out. So one of the limitations of current imaging technology is that the standard cancer imaging techniques like the PET scans and the CT scans, they don't have the resolution to look at the single cell level and see in real time what's happening. You have to usually wait until the tumor reaches a size that's detectable by these imaging platforms and softwares. So what I did is I developed a system where we can look at blood vessels in real time. And so what we're seeing here is an image of a blood vessel. It's fluorescently labeled, so all the endothelial cells and the blood vessel cells are marked in red, and then injected cells that are human cells injected from cancer patients or from cancer cells in the blue here. And so what I used is a type of microscopy or a microscope called a light sheet microscope. This allows me to take very thin sheets or layers of microscopy images and then stack them on top of each other to create a 3D reconstruction of the image where I can take a deep dive and a comprehensive look inside of the blood vessels in real time and take a deep dive and kind of swim through the blood vessels and see what's happening, where these cells are, how they're exiting, and then what the effect and the impact is after they leave. So I'll give you a quick example. This is a video. So the blue cells that you see are human cancer cells, and then the red is a blood vessel or endothelial cells that make up the blood vessel. Now you can see this is a cell that is leaving the inside of the blood vessel and entering the tissue surrounding it, which will then most likely be the type of cell that would form a secondary tumor at this site. So it gives us the ability to look at the single cell resolution of how these cancer cells are exiting and spreading throughout the body, specifically how they're exiting blood vessels to form secondary tumors. So observing the cancer behavior. So what this allowed us to do, it allowed us to observe specifically how the tumor cells travel throughout the body or throughout the circulation, capture exactly how they exit the blood vessels, and then analyze specifically which cells grow secondary tumors and which ones don't. So like I said, this is a highly inefficient process, so only certain or some cells grow and form these secondary tumors, not all of them. So we really need to understand which ones do, how they're doing it, and why. And so this allowed us to observe it in real time rather than looking at some of the current limitations, which is imaging a tumor after it's already reached the size that the imaging software is able to image it. This is actually able to look at the cells in real time as they're spreading throughout the body. And what we discovered is that both single and the cluster cells, they do exit and they do survive, both of them, but it's the clusters, the groups that actually have a much higher metastatic potential or much larger chance of growing once they exit the blood vessel compared to the single cells. So this led to a new understanding. We named this or dubbed it, it was dubbed the cancer X's hypothesis, that proposes that the tumor cells are able to travel throughout the circulation while maintaining their multicellular phenotype. But not just that they're actually able to exit the blood vessel and then form new tumors, all while maintaining the multicellular phenotype. So it was previously thought that, again, they had to break or disassociate before exiting or some point throughout the whole process. But what we discovered is that they're able to travel from one primary site, travel in the circulation, exit, and form new tumors, all while maintaining their multicellularity, and that this actually is not just a byproduct of something, but this actually allows them to form secondary tumors at a much higher rate. So it's estimated that more than 90% of all secondary tumors are formed from the clusters rather than single cells. And so what we saw is that, again, this video illustrates what I was talking about. Both the tumor cell, single cells, and clusters, they can exit the blood vessels, but it's the clusters of cells that have a much higher propensity to grow and form these secondary tumors, which is really key to what we're wanting to figure out with cancers, not just what's happening, but which are the ones that are causing these deadly secondary tumors. So we took it a step further where we wanted to look at we saw the visualization of this process, but we wanted to take a deeper dive into the molecular side of things. So what I did is I developed a system where we're able to have a photoactivatable engineered cells. So these are human cells with a fluorescence protein that is photoactivatable. So what that means is the cell might be producing a green fluorescence protein. It's activated by UV, and then it turns to, say, a blue or another fluorescence protein, specifically after photoactivation. So what we did in this system, we had the blood vessel, or we imaged the cells as they were exiting the blood vessel, and then we targeted them using a specific UV activation to target only the cancer cells that exited as clusters through this method. And so after that, we were able to isolate these cells using fluorescence-activated cell sorting, or FACS, which just allows you to take specific fluorescence and then sort it by the fluorescence intensity or type. And so we were able to take specifically only cancer cells that successfully underwent this process and then do some genetic analysis. And what we found is that they had several key pathways that were modulated differently, things like cell adhesion, vascular remodeling, and several other things. The most important one, or insight, is that this is a transient process, so it only occurs at specific times. So if we're looking at cancer cells in the normal capacity where they're growing and then we image them, we're losing a lot of this genetic information that we normally have. So shifting to the kind of the next phase of cancer and where this is taking us, the next era of cancer research is going to be taking a lot of machine learning and AI algorithms and applying it to these circulating tumor cells to decipher and decode what's happening in real time with a lot of the information that we have in genetics. And a lot of the work that I'm doing now has shifted into the clinical space, where I'm working and focusing more on clinical trials and clinical research. And this area is going to be a lot more precise with the advent of a lot of the technology, and there's going to be an integration of genomics, bioengineering, and then having a more personalized research plan for each individual, almost treating each disease as its own personal separate disease rather than a broad disease. And then two, next generation therapies where we're going to have things that disrupt, like I mentioned, it's not the cancer itself that is deadly, it's when it spreads to multiple points. So we're going to have systems that are more targeting preventing cancer from spreading and then stopping it sooner rather than later. So the big picture takeaway, cancer doesn't spread randomly. Blood vessels actively help tumors escape. The cancer cell clusters are the most dangerous travelers, and the tumor cells that form new tumors are unique. And the most important one is that understanding how cancer therapy or cancer escapes, it gives us a unique perspective and a therapeutic strategy to now target to reduce metastasis and save lives. Thank you, and I'll end there.
It's a great honor to be here and tell you about our contribution to computational microscopy. And let's start. So our story begins with super resolution microscopy, and specifically a method called localization microscopy. So to give some motivation, let's say you have a sample you want to look at with a microscope, fluorescence microscope. In this case, you see these microtubules. So it's a cellular structure, but your resolution is insufficient, and you want to see more intricate detail than the best resolution the microscope allows. Now, the fundamental limit on resolution comes from physics, from the wave nature of light, and it's about half the wavelength. So you get, even with the best optical microscope you can buy, a resolution of 200 or 300 nanometers. If you want to do better than that, then localization microscopy is a technique that was developed for that. And the idea is, instead of capturing a single image, you capture a set of frames, a sequence of frames. During these frames, the molecules, individual molecules, are blinking, turning on and off. You take each one of these blinks and you say, I know this is only one molecule, so I can localize it, and you put a dot on the right side. And slowly but surely, you get a super resolved image of your structure. So there's a tenfold increase in resolution. And this is now a well-established technique. It's been the subject of the Nobel Prize in Chemistry 12 years ago. What we do in the lab is we are taking this to the third dimension, which means if you have a volumetric sample, and any sample is volumetric because a cell, even though it appears thin, it's actually, it could be 10 microns thick. The depth of field of a high NA microscope could be 1 micron or less, so things go out of focus, and this is going to be a problem if you want to image something with a volume. So to demonstrate this, this is a model of a microscope. You see a point source moving in and out of focus, and the effect on the camera is that when it's in focus, you see a nice tight spot, but when it's out of focus, it becomes a blur. And any kind of three-dimensional imaging you want to do with such a microscope is going to be a problem. And the way we circumvent this is we add an optical element to the microscope. It's this element we call a phase mask. What this does, it modifies this shape that the point source generates on the camera, which is called the point spread function, the PSF. And now it looks like this shape when it goes in and out of focus. So it's no longer a blur. It looks like something strange. But it turns out that this is the optimal way to encode the 3D position of a point source in a microscope. Given a limited number of photons, you can ask, how do I want to spread these photons on my camera, such that I'll have the best possible precision when I try to determine the position of this point source. We use tools from estimation theory to come up with this solution. Specifically, we're maximizing the Fisher information of the system. And then we put this phase mask, and now what you can do is 3D localization microscopy with this very good point spread function. And this is what you see here. Each shape here that's blinking is a single molecule, but the shapes appear different because they are at different depths, and each depth is encoded differently because of the phase mask. So there's no scanning going on. It's a staring system, but you get volumetric information. So in gray, you see the experimental measurement, and in purple, you see what our neural net that we trained to decode this information understands. So you see there's a good fit, and at the end of this experiment, you get a 3D volumetric reconstruction at super resolution. So the resolution here is about 50 nanometers in XYZ. I want to stress that the only way this was possible is thanks to neural nets. There was no algorithm that could fit all these overlapping shapes that I could ever find. So this is only possible in recent years. Okay, now the question is, can we design a better 3D PSF for this specific problem where you have a high density of emitters and you know they're going to overlap, and overlapping is a severe algorithmic problem. And the reason this is important is because, as you noticed, acquisition takes time. To get a single image or a volume, you need to take a whole movie. So if your sample is dynamic, or if you just want reasonable throughput in your experiment, you're going to have to squeeze in more molecules in a given frame, and this is going to cause overlap. So high density. So a long story short, how do we design this better, or hopefully the best point spread function for this problem? And the technique is interesting because what we did is we let the neural net guide us. So as the neural net is learning to decode this information, it's also learning how to encode it. So it's learning the optimal phase mask that would yield the best result. This is called end-to-end optimization. And you see the phase mask being learned here, slowly, and this is the phase mask, and this is the 3D point spread function that corresponds to it. And we can show that it's better in precision. Okay. The holy grail in super resolution microscopy is to do dynamics, because any super resolution technique takes time, whether it's localization microscopy or scanning techniques. And time means it's difficult to image dynamic objects like live cells. So if you want to do live cell super resolution, you're going to have to do something more clever. Here's an example. This is a blinking cell. These are microtubules, but the cell is moving. The reason the cell is moving is actually, okay, the camera is moving, but that's to demonstrate the point. So if you just sum all these frames and localize them, you're going to have motion blur because you're summing stuff that moved, right? So you don't get a clear picture. So can we do super res from blinking move of super single molecule localization microscopy video to super resolution video? So the answer is yes, if you allow some prior information to be used. So for example, if you know you're looking at microtubules, you can train this recurrent neural net that takes into account long-range temporal interactions and recover the dynamics. This is a clearer result where you see mitochondria blinking, and this is the reconstruction from this movie. So here we're venturing into this dangerous realm a bit of a lot of prior information. So you have to be careful and validate these methods because neural nets, as we know, can hallucinate. Nevertheless, this was validated pretty strongly. Okay, so an elephant in the room is the question whether people can actually use these tools we're developing. So our customers at the end of the day are biologists. We want them to be able to use these tools. So, but biologists have real problems with their biology, and they don't need to be spending time on installing packages and building optical setups. So how do we make it happen? We need accessible hardware and software. I won't talk about software, but briefly, we're making all these algorithms one click, so anyone can use them. But the hardware is still a problem. So to understand the hardware issue, at the heart of this technique is a phase mask. What is this phase mask? It's a dielectric structure. So it's a piece of dielectric material that has different thicknesses, and those, as you shine light through this, the wavefront is being modulated by this landscape. It's very difficult to fabricate something like this. We need photolithography because it's very tiny resolution that's required. Because the wavelength of the light is hundreds of nanometers. If you want to do wavefront shaping, you need to get tens of nanometers precision. So anyway, it's very hard to fabricate, and we realized to make this, to democratize the method, we must come with a simple way of fabricating phase mask. And our realization was this: light doesn't care about physical distance as it propagates, it cares about optical distance. So what this means is, if you take a thin piece of glass and shape this plane wave by it, and on the other hand, you take this very thick piece of polymer immersed in another polymer or attached to another polymer with very close refractive index, you get the same optical effect. But the bottom one is much easier to fabricate. We can 3D print these templates, and we can now do it in the lab. We hold it in our hands. Nowadays, we print these things in 10 minutes, which is orders of magnitude faster and cheaper than you would do it with lithography. So this is based on near index matching. Okay, and we've done all kinds of demonstrations with this, different phase masks and different results that I don't have really time to show you. We also fabricated vortex plates, so we can fabricate all kinds of optical elements now, not just for microscopy. And these are achieve state-of-the-art results when you compare to the electric masks done with photolithography. The last thing I want to show you is that we can implement this in high-throughput microscopy, which is very useful. We just stick this mask in an objective of a high-throughput microscope, and then we can capture hundreds of images or volumes and train neural nets based on this experimental data that give you snapshot 3D information. So this is a single cancer spheroid imaged with our phase mask, single shot, and the network allows us to recover the 3D structure. And this is very important if you want to do high throughput research, because you want to screen many drugs, or etc. Okay, I'm out of time, so thank you very much for listening.